Our next student is Ashley Cousin from Benjamin School. The thing that really made Ashley stood up out of all the 90 applications, and I remember hers from the beginning. And also during her interview, she's the only one that in the application was like, I want to work in the Bolton lab because I had the opportunity to volunteer with kids with autism spectrum disorder. She's volunteering as a tennis teacher, and she's like, I want to understand more because this disease has just one name, but actually is a spectrum. There are a lot of things in this disease, and I want to understand more. And she was so convinced that she made it, and she did an awesome job during the internship, and she's going to tell you more about what she studied involved with autism spectrum disorders.
Hello everyone, my name is Ashley Cousin, and this summer I had the amazing opportunity to be a high school intern in the Bolton lab. During these 6 weeks, my project was exploring the morphological abnormalities of amygdala neurons in models of autism spectrum disorder. First, what exactly is autism spectrum disorder, or ASD? So this is an umbrella term, and as the name suggests, it is a wide spectrum. It affects every individual differently. However, there are the common symptoms of impaired social interaction, language and communication difficulties, and repetitive behaviors. But the trouble is there is no one single autism gene that can be pointed to the same cause. It's related to a variety of genetic and environmental factors. One of the main brain regions involved in ASD is the amygdala. The amygdala is important in processing anxiety and fear memory. Any abnormality in function of the amygdala can lead to anxiety or other symptoms characteristic of ASD. The amygdala is in this region, and this is a diagram showing the central and the basolateral amygdala. This is also an image showing an example of the amygdala. If you are alone and crossing the street, you might be fearful, but if you see that you're with an adult, and last week you remember crossing the street safely with an adult, then you might not be scared. And that's all in the amygdala, fear or no fear, as it receives inputs from other various regions of the brain. Like I said before, ASD can be caused by a variety of environmental and genetic factors. Some of the genetic factors can be organized into families. There are the genes that code for proteins related to the formation and function of synapses. There are genes that are related to ion channels, and there are genes that are related to intracellular processes. The 2 genes that I am studying during this project, PTEN and TBR1, are both in this family of intracellular processes. The first gene I'm looking at is the phosphatase and tensin homolog, or PTEN gene. So this is a regulatory gene, it's a tumor suppressor gene, and so if it's knocked out, or if there's an issue with it, it could cause a growth issue where cells become overgrown. The behavioral phenotype seen when this gene is knocked out is that in behavior tests, knockout mice demonstrate more fearful behavior. This is an ASD-like phenotype. For this project, I'm looking at the dendrite morphology in somatostatin cells. So I'm looking at differences in the size, length, etc. of the dendrites, where the cell receives its inputs, in somatostatin cells. Somatostatin cells are a type of interneuron in the central amygdala. The method for this portion of my project is first the cells were patched and filled by my mentor, Tim. Thank you, Tim. In this process, he is listening in almost to the cells, and while this is going on, the cells are being filled with biocytin. During the next stage, I do an immunostain on the slices. In this, an antibody binds to the biocytin, and the result is that the entire cell is fluorescent. Next, I mount the tissue and take an image at the confocal microscope. The next thing that I do is I upload the 3D image into a special program called Neuralicida, which allows me to trace out the dendrites and then analyze them. These are the resulting traced images. So these are the PTEN knockout, and this is an example of the wild type. I did a shoal analysis, resulting of which is that there's these rings. each of which is 10 microns larger in radius than the previous. As you can see, there are many more rings needed to encapsulate the entirety of this cell as compared with this, which means that the PTEN cell is much larger than the wild type. That's to be expected because, like I said before, it's a regulatory gene, and so when it's knocked out, cells can become overgrown. This graph also shows that result. The wild type, represented in blue, don't extend as far as the knockout, the wild type, represented in black, as you can see, don't extend nearly as far as the knockout, represented in blue. The knockout cells, on average, extend around 370 microns from the soma, or the cell body, but whereas the wild type only extend on average 220 microns. As you can see, the dendrites in the PTEN knockout extend nearly twice as far as the wild type ones. This is a huge difference. The next analysis I did is on dendrite intersections, and I found that closer to the soma, the cell body, so in this area, the wild type dendrites had more intersections. By intersections, I mean how many times the dendrites cross those rings you saw. And as you can see, the wild type, once again represented in black, has more intersections in this area closer to the soma than the PTEN knockout, represented in blue. Intersections is a measure of complexity. So this graph suggests that the wild type dendrites branch off earlier than the PTEN dendrites. The final analysis I did for this portion of the project is a soma volume. So this is the cell body volume. Once again, the wild type is represented in black and the PTEN knockout is represented in blue. And what I found is that the soma of the PTEN knockout is much larger than the wild type. This again is to be expected as PTEN is a regulatory gene. The next part of my project was looking at the T-box brain 1, or TBR1 gene. This gene is a transcription factor, so it is responsible for turning on and off other genes. When this gene is knocked out, the resulting structure issue is that there's a projection issue. By projection, I mean the axonal projections you can see here, which is how the cell sends out its outputs. The behavioral phenotype observed is that the TBR plus-minus mice show impairments in aversive learning and social interactions. That again is an ASD-like phenotype. For this portion of my project, I'm looking at the cell body count in pyramidal neurons in the amygdala. These are images of the wild type and TBR plus-minus cells in the amygdala, but looking at these images, you might not be able to see that much of a difference in the amount of cells. This graph clearly shows that. As you can see, the TBR plus-minus cells are represented in gray, and the wild type are represented in yellow. For this, I looked at a 200 by 200 square area of the amygdala and a 500 by 500 square area of the amygdala. And in both of these areas, there were fewer cells that projected to the prefrontal cortex in the TBR plus-minus as compared to the wild type. So, what does this all mean? Why is it important to study morphological abnormalities? Why do we care if in the PTEN knockout the dendrites extend nearly twice as far? Well, it's important because any changes in structure can lead to consequences for function. In the case of PTEN, if the dendrites are too long, then they might not carry the message as well, which could result in the observed phenotype. And in the case of TBR plus-minus, if there are fewer cell bodies projecting, then that means there are fewer inputs and outputs, which could result in the trouble the mice have learning. I'd like to say a huge thank you to my mentors, Tim and Ingo, and also the entire Bolton Laboratory for hosting me during these 6 weeks and being so supportive. I'd also like to thank Ilaria for all her help during this process, especially in preparing this presentation. And I'd also like to thank the members of the Brain Trust because your guys' donation helped give me this amazing opportunity.
Question for Ashley? We start here. Yes.
Were the treated cells done in vitro or at what point were they treated?
Oh, the question was at what point were the cells treated? In regards to the PTEN, the knockout was done genetically, so the mice are born with it. Any other questions?
This is beyond, but like for what's next, having identified this, is there some thought that there's some treatment that might come out of this?
Okay, so the question was sort of what's the next step? Like, is there any treatment that's going to come out? So the important thing before any treatment is to completely understand what's going on structurally and functionally, but I believe the next step that my mentor will then be working on is to rescue this behavior, but that's in the future. It's important to first understand what's happening, like structurally.
I was wondering what was your sort of aha moment, were there surprises along the way that were unexpected?
The question was, what was the aha moment? For the aha moment that I saw was when this graph was made comparing the dendrite length, because as I'm staining them and taking focal images or tracing the dendrites, it's hard to see the difference of the two cells. They both look around the same size, even when I trace them. But when I did the analysis and made this graph, I was like, my mentor and I were completely amazed because this is a huge difference. And yeah, because if you look at these images, they don't look that different here until you do this analysis.
Andre?
So you mentioned in the discussion that there could be a correlation between the morphology and the physiology. Did you see anything like this? So in terms of like increased IPSCs, EPSCs in the
So the question is that, are you referring to the like electrophysiology or the behavior? Okay. So the question is about the electrophysiology of these cells. You'll have to ask my mentor, Tim, to be completely sure. He's the one doing those experiments, but I believed there was a difference, that the PTEN didn't carry the message as well, perhaps because they're too long.
Any other question? Thank you so much.